Hello, this is talk number 26 in a series from the 7th edition of Principles of Biochemistry by Albert Lehninger. Today we'll be discussing amino acid metabolism, production of urea, and the urea cycle. Amino acid oxidation, production of urea. These are the primary energy source for carnivores, amino acids, obligate protein consumers. Rare use of this pathway in plants. There are three circumstances for energy use. One is that these amino acids are not necessary for protein synthesis. Also, the surplus of amino acids are encountered, consumed, or catabolized, and in starvation or diabetes mellitus, amino acids are used as energy sources. And the process of developing a certain starvation appearance where amino acids and proteins are actively being utilized for catabolic purposes, we refer to that as marasmus. Generally, amino acids lose the amino group and become alpha keto acids. They're oxidized and converted to glucose or carbon dioxide via the citric acid cycle. Some of these amino acids are more readily transformable to that cycle than others. Will be discussing that. For the biological fate of the amino group, we consider that the amino group, which is toxic, will be converted to urea or uric acid. Of these, the most readily converted amino acids to citric acid cycle intermediates are glutamine, glutamic acid, alanine, and aspartate. In the stomach, gastrin stimulates hydrochloric acid, which digests protein, and pepsinogen is converted to pepsin, and pepsin will hydrolyze the protein adjacent to leucine, phenylalanine, tryptophan, or tyrosine. Secretin is secreted into the small intestine that stimulates bicarbonate release, neutralizes the contents, and then there are numerous pancreatic enzymes released. They're inactive precursors which are activated. They further hydrolyze protein such as chymotrypsin and trypsin. These are transported into the small intestine epithelium. There are catabolic steps. The amino acid group is removal is done in the liver by transaminases. There are two broad ones in the liver, and the amino group is transferred to alpha ketoglutarate. Two types of the transaminases, or alanine aminotransferase and aspartate aminotransferase. And this is the means of action here. So you've got the amino acid in this case. You have pyridoxal phosphate, which is an internal aldamine form, and it's present on the enzyme. This is a vitamin. This is vitamin B6. So you have you've got this the enzyme right here. You've got this moiety here, and you have a Schiff base intermediate that's formed, and that Schiff base is present at this point. So so we're looking at the end point of the pyridoxal phosphate right here, occurring right there. This is the Schiff base. You have an abstraction of proton. This double bond is shifted over to this point with the proton leaving, with the base picking this up. This leads to a quinoid formation right here. This is a quinoid intermediate, and the electron pair picks this up, picks up a proton here. You get this rearrangement of the bond at this point, with the double bonds remaining here. Then the Schiff base is hydrolyzed right here, and it forms a keto acid from the original amino acid. This is a transformation that occurs, and you get a release of pyridoxamine phosphate. quinoid intermediate that I reference here. This is the transformation that occurs right here, and the enzyme comes in and picks up the PLP right here, and you have the amino acid produced at this point. So this is one of the transformations that occurs with quinoid intermediate, and also you have the shift base present, and the shift base is an external aldamine, and you can see the quinoid intermediate here, picking up the proton, and then there's a shift of the double bond. The amino group is broken off. This is hydrolyzed, and pyridoxal phosphate is released. So this is the means by which you can transform amino acids into entities that can enter the citric acid cycle here. The ammonia is collected in the liver as the amino group of L-glutamate. This is oxidized by L-glutamate dehydrogenase, so you get a reduction of NAD, and this is forms this enamine link here. This is hydrolyzed, and you get alpha-ketoglutarate and free ammonia. This is another representation of L-glutamate here. ATP is the energy requiring steps. You get this entity formed at this carbon here, ester linkage, and then ammonia is comes in. You get amination of this with release of phosphate ion. This is L-glutamine right here, and then the ammonia is cleaved off from this point as and enters the urea cycle. So you're trying to get this ammonium ion right here, and glutamate is the end product. So this is how you can recycle ammonia through a ATP linkage here, ammonia placed in this point here, and then released as a cycle. The ammonia enters the urea cycle in the liver. Alanine transports ammonia from the muscle to the liver. Ammonia is a toxin, especially to the brain. It competes with potassium for cell transport and does cross the blood-brain barrier. The urea cycle, birds and reptiles excrete uric acid, generally in crystalline form. Very little water needed by these animals. Terrestrial animals excrete urea. There are 5 steps. The first of these is ammonia is transferred to ornithine to form citrulline. Ornithine is synthesized from glutamate and is a carbamyl phosphate intermediate. Second step, aspartate amino group is transferred to citrulline, forming arginosuccinate. Arginosuccinate is cleaved to arginine and fumarate. Fumarate is converted to malate, and this enters the citric acid cycle. And arginine is cleaved to urea and ornithine. Schematic of it right here. So ATP is here, there's bicarb. So again, we've activated the bicarbonate phosphorylation right here. It's an anhydride linkage, which has an energy cleavage point here, and you get ammonia displaced in the phosphoryl group, and you get carbamate ion right here. And then this carbamate ion is phosphorylated, and this yields carbamyl phosphate, which is here. So you get attack at this point, and phosphate group attached to this point. So you have carbamyl phosphate right here. Also at the same point, you've got this ATP with citrulline bonding. This is another manifestation. So you get phosphorylation present or release. You get AMP adduct through this carbon here, and then there's rearrangement of this molecule with the base pair at the amine group here, kicking in and kicking out. AMP is released, and you get attachment of aspartate to this citrulyl AMP. So the presence of this bond drives this reaction here, and you end up getting what is arginosuccinate here, where you've got aspartate amino acid here, and you've got the amino group from citrulline here, with bonding occurring through this area here. And these are the side chain of citrulline over there. The urea and citric acid cycles are linked chemically. They're regulated through the synthesis of urea cycle enzymes. Carbamyl phosphate synthetase is regulated by N-acetyl glutamate. One molecule of urea requires 3 ATPs, 2 for the carbamyl phosphate and 1 for arginosuccinate. Amino acid degradation, there are 20 pathways, 6 products, and about 10 to 15% of human energy production is geared towards that. So here's the citric acid cycle. We've reviewed this: alpha-ketoglutarate, succinyl-CoA, fumarate, oxaloacetate, picking up acetyl-CoA, forms citrate, isocitrate. Acetyl-CoA can go on under certain conditions of duress, starvation, to ketone bodies, along with acetoacetyl-CoA. And these amino acids here can be broken down to acetoacetyl-CoA. These amino acids, acetyl-CoA, these amino acids can give rise to pyruvate, asparagine, and aspartate to oxaloacetate, and phenylalanine tyrosine to fumarate, and so on. Now, if you look at the structural similarities between the precursor molecules and the product molecules, you can see at a glance how you can get from one of these to the other. So it should not necessarily be particularly difficult to be able to characterize not necessarily the amino acid, not necessarily the aromatic moieties, but the base structures. You can determine how they would fit into these products here. Again, these are the ketogenic pathways here, again, under conditions of starvation or other issues. And then the glucogenic pathways are here, because these would ostensibly go back and break down products of glucose, so they would be related in that regard and be an energy-forming thing. So ketogenic amino acids form ketone bodies, so gluconeogenic amino acids form constituents of the citric acid cycle. And the cofactors involved are biotin, S-adenosyl, methionine, tetrahydrofolate. All these are involved in one carbon transfer. So biotin has this structure here. We talked about the structure of biotin using the amine group as a carbon transfer entity. S-adenosyl methionine is the amino acid methionine. This is the sulfur group that's a characteristic of that, and it is bound directly to the C5 of an adenosine molecule. This is adenine here, this is adenosine. The folate is here. There's a delta or 6-methyl pterin right here, and this part is the P or para-aminobenzoate moiety there, and glutamate is here. So this is tetrahydrofolate, which is a component of folic acid. Again, this is a one carbon transfer, and is again one of the a nutrient that's required in the body. Branched amino acids, branched chain amino acids are degraded in non-liver tissues. Energetically, S-adenosyl methionine transfer is most favorable, and there's a host of metabolic diseases that originate with amino acid metabolic deficiencies. And here's a list of them. So there's the incidence of per 100,000 births. These are highly uncommon, but you want to be able to catch some of these. So there are laws on the books to assess neonates for phenylketonuria. A lot of these are much more uncommon, and generally, if you have failure to thrive or some other neonate who is developing marasmus or kwashiorkor, where you have the dilated abdomen, you can do screening to determine if there are increases of amino acids or precursors that are occurring. So albinism is, for example, melanin synthesis from tyrosine. Defective enzyme is tyrosine enzyme, tyrosine 3 monooxygenase, and these are the symptoms that occur as a consequence of that. So you find out where the defect, what the symptoms are. You do the tests, you determine what buildups of precursors there are, and then you can assess that these are the enzymes. These are usually autosomal recessive, whereas you get a gene from each parent, and this is a condition that occurs. Maple syrup urine, the urine literally smells like maple syrup. A lot of these have mental retardation and early death as consequences of this. Others you can live with, such as albinism, but it does have a distinctive clinical appearance. Again, phenylketonuria, PKU, is the disease is effectively screened, but you want to catch these very early if possible. And that's it for the talk. Thank you very much.